microplastics are contaminants of emerging concern that are increasingly ubiquitous within the natural environment. They can exist as either primary microplastics, which are manufactured to be less than 5 millimeters in diameter, or secondary microplastics, which are much more environmentally relevant, as those are the broken down plastic pieces from larger microplastics. One of the biggest concerns with microplastic as a pollutant is their ability to absorb lipophilic organic compounds and then act as a vector of exposure to organisms. PAHs, or polycyclic aromatic hydrocarbons, are one of these organic compounds that can enter the environment through events like oil spills. Microplastics are also highly diverse in both polymer type and shape. So for the purposes of this study, we looked at 3 different microplastic types. The first being a primary microplastic, which is what's most commonly used in toxicological studies, which are polyethylene beads, and then 2 secondary microplastics, which are polyamide fragments and polyacrylic fibers. Additionally, we use the hard clam as our study organism, as they are sessile filter feeders and more likely to be exposed to these contaminants. Now, while oxidative stress is an endpoint that we're really interested in, as both PAHs and microplastics have been shown to independently induce oxidative stress in organisms, for the purposes of this talk today, I wanted to mainly discuss mortality data. So we conducted a 96-hour static exposure where we exposed organisms to 6 different oil sheet thicknesses, as well as each of our different microplastic types and a non-microplastic treatment. At the conclusion of this exposure, we then collected our organisms and assessed them for mortality under a microscope. From here, we conducted a generalized linear model using both row and replicate as random variables in the model, and we were able to pull up these results right here. So as you can see from this graph, we saw a decrease in percent of survival as oil sheet thickness increased when no microplastics were co-exposed. However, when we introduced the primary microplastic beads, we actually saw an increase in survival up to 95%. So this is particularly concerning because, like I mentioned before, most microplastic studies are using these primary beads. So we might not be seeing as much risk with microplastics in existing studies as there actually are in the environment. Our secondary microplastics, fragments and fibers, we saw an increased rate of mortality than with no microplastic present. Our fragments saw an increase of 3% and our fibers an increase of 32%. We believe that this is likely because the fragments and the fibers have a higher surface area to volume ratio and therefore can transport more of the pollutant per particle, but also absorptive capacity of the polymer used to be playing a role, and we hope to confirm this further with